looks like we got a fair number of folks, so I'm going to go ahead and get started. I'm Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Happy New Year. Welcome to the first webinar presented by ASCCT together with the European Society for Toxicology in Vitro. This is our first one for 2022. I would encourage you to go visit our websites if you're new to either of our organizations to find out more. And please consider either signing up for our email list or becoming a member to support these programs and to find out when you can register for the next one. Both of our organizations are focused on supporting and promoting computational and in vitro toxicology fields and scientists and students that work in them. We offer training and education, meetings, discussion forums, information, and aim to support early career scientists in their endeavors. So this webinar is recorded like all of our webinars. The recording will be posted on the ASCCT website a few days from now, along with the slides. Your lines are muted in the webinar platform. You can ask questions of the presenters in the Q&A feature at the toolbar on the bottom of your screen. The main Q&A will be after both speakers have finished. However, please enter your questions at any time they occur to you. And if there are quick clarification questions that we can address, we'll do that after the first speaker is done. If you have a technical issue or you just want to put a comment out to the rest of the participants, feel free to put something in the chat, but please do put your questions for speakers in the Q&A. I just wanted to give you some information about some activities STIV has going on. Both of these activities are open for registration. First of all, there is a applied in vitro toxicology course for 2022, that should say, that's going to be held in June. You can go to the STIV website to register for that. And then the STIV Congress is going to be held in November of this year in Barcelona. And so we'd encourage you, abstract submission is open. So we encourage you to go to the website and find out more about that and participate. For the next few months, we're going to be offering some more webinars featuring some of the early career scientists and students that have won awards last year from STIV and ASCCT. And we have some other topics lined up for you, including mixtures toxicology, NAMs and risk assessment, serum-free cell cultures, and lung models, among other things. So make sure to visit our website and find out when those are announced. And now I'll bring you to our current program for today. We're going to be talking about advances in genotoxicity using reconstructed Skin Comet and Micronucleus assays. Our presenters I'll introduce now, and then we'll hand it over to them. So Dr. Kerstin Reisinger received her PhD from the University of Frankfurt. As a biologist and registered toxicologist at Henkel, she has worked on the development and evaluation of in vitro methods for safety assessment of substances, including cosmetics, for several years. Her work focuses on genotoxicity as well as skin sensitization and eye irritation. Dr. Reisinger is a member of several working groups and associations, such as the Cosmetics Europe Genotoxicity Task Force. And Dr. Stefan Pfuhler received his PhD in biology from the Department of Pharmacology and Toxicology at the University of Ulm in 1997 and joined his current employer, Procter & Gamble, in 2000, where he serves as research fellow in global product stewardship. In recent years, Dr. Pfuhler's research focused on alternatives to animal testing and genotoxicity testing strategies, and he leads validation efforts for three-dimensional human skin-based genotoxicity assays. He's chairing Cosmetics Europe's Genotoxicity Task Force since 2002 and serves as co-chair of Hesse's Botanicals Consortium as well. All right, Kerstin, I will give the floor over to you.
Thank you very much, Christy, for the nice introduction and for giving us the opportunity to talk about two new in vitro assays, which are based on skin tissues. And I saw the number of all the colleagues of you who are joining, and I would very wish to know where your computers or tablets are set, and whether I should say good morning or good afternoon or good evening. In the next minutes, I like to share some background on the concept of why using 3D skin tissues for genotox testing before I add details on the reconstructed Skin Comet assays. I will then hand over to Stefan, who will talk about the reconstructed skin micronucleus assay and will add details on the strategic fit of both assays for regulatory decision making. When we look at the regulatory landscape and the European Union, we see a growing number of legislation which restrict or ban animal experiments, and especially the EU Cosmetics Directive. But this development, that this tendency is not only true for the European Union, but there is a growing number of regions around the globe which foster or ban in vitro only approaches for the safety assessment of cosmetics. The good news is that the classical in vitro genotox test battery is known to be quite sensitive for the identification of in vivo genotoxicants. However, the test battery approach, the combination of several assays leads to a reduced specificity, so the capacity to correctly identify non-genotoxicants, and that leads to a clear fraction of misleading positives. One reason for this was identified in the type of test system the classical in vitro genotox assays are using, as they are based on 2D cultures, which are limited in reflecting the in vivo situation. It's different with 3D tissues. Their key parameters are similar to in vivo test system in terms of proliferation, morphology, or function. And therefore, they are suited as complex test system for higher tier testing, as they also close two gaps 2D cell cultures may leave. As 3D tissues can mirror the route of exposure, and they have an intrinsic metabolic capacity. And that was the background, the starting point at which Cosmetics Europe started to define its animal-free strategy for genotox testing. And I will not go in any detail, but just pick one pillar, the development and validation of new in vitro assays, and pick from this pillar the two projects working on the dermal route of exposure. And this is the reconstructed skin Comet assay and the reconstructed skin micronucleus assay. Classical readouts combined with the Fenian full thickness tissue for the Comet and the epiderm model for the micronucleus test. These were the first two in vitro options for genotoxicity assessment of the dermal exposure route, a route relevant not only for cosmetics or household products, but also for pharmaceuticals, agrochemicals. Both tissues are based on primary p53 competent cells of human origin, so a normal cell cycle control can be anticipated. In addition, the gene protein expression pattern is similar to human native skin, especially the enzyme pattern related to the xenobiotic metabolism. And therefore, the utilization of external metabolizing system like the red liver S9 mix is not required. And last but not least, these tissues are for sure topically treated, reflecting the barrier function of the skin and allowing application of concentrations which may be toxic to 2D cell cultures. Both tissues have been selected from a variety of tissues based on assay-specific performance criteria. And that resulted for the Comet in the selection of the Fenian full thickness model. Full thickness model means that it comprises epidermis made of keratinocytes and a dermis made of fibroblasts which span into collagen matrix. As I mentioned, that the tissues were combined with classical readouts, and we present them together as a package, as both assays allow addressing all three types of DNA damages, which needs to be addressed for regulatory decision making. The assays are not intended as replacement of the classical in vitro assays, but to fuel the toolbox of the second-tier testing to follow up on positive findings from the classical testing. But Stefan will add more to that aspect. This package of the two assays, the idea, the concept has been presented several times and were reviewed by external experts, which are shortly listed on this slide. And I just want to highlight the IWGT meeting in Tokyo in 2017. IWGT is a global and international working group of gene tox testing, which meets every 4 years. And the expert in Tokyo summed up after the review that 3D tissue models offer indeed a more in vivo-like behavior for key parameters, and therefore they provide a valuable complement to the classical 2D cell culture-based assays. And based on that, they can be used as second tier, confirming the intention of the Cosmetics Europe Gene Tox program. And with that introduction, I like to come to the Comet assay in more detail. The Comet assay, as you know, does not rely on proliferating cells, and therefore allows the investigation of DNA damage in any cell culture or tissue which can be subjected to single cell isolation. And that makes the Comet assay a widely used method for the detection of DNA damage in gene tox testing, in human biomonitoring studies, or environmental monitoring. And the increased relevance of the assay has been documented in 2014 with the release of an OECD testing guideline for an in vivo mammalian Comet assay, the alkaline version of the Comet assay. And this version detects a wide range of DNA damage, including modification that leads to gene mutation. And recent publication also confirms this and proves the high sensitivity of the in vivo alkaline Comet assay for carcinogens that show gene mutation activity, and the capability of the alkaline Comet assay to pick those carcinogens is similar to the predictivity of the TGR assay, the transgenic rodent gene mutation test. And also the Skin Comet assay uses the alkaline version of the Comet assay. So we start here with the exposure of the skin tissue, and after the treatment period, first epidermis and dermis are separated using an enzyme, and then the single cells are isolated separately for epidermis and dermis by cutting the compartments using the so-called mincing procedure, which results in a mixture of single cells and single nuclei. As mixture is resuspended in low melting agarose and transferred to a glass slide, which is the agarose is then led to solidify to allow the incubation of the slide into a lysis buffer, which disintegrates the still existing cellular membrane and to salt out the proteins, especially the histones. And subsequently, the still supercoiled DNA is let unwind under high alkaline condition. And that is the prerequisite that the DNA can migrate afterwards in an electric field still under high alkaline conditions. And when the DNA has been damaged, then the single DNA fragments can migrate in the electric field, but also relax DNA loops. And then the slide is prepared for a subsequent staining of the DNA, which is then analyzed semi-automatically. And the software helps to calculate the fluorescence intensity in the comet tail built by migrated DNA in comparison to the comet head, which is made of DNA which were not able to migrate under the used condition. That is still, even if it is semi-automated in the analysis, can be quite time intensive. So there are possibilities to further automize this. And Procter & Gamble, Stefan's laboratory, has put effort into that with the Comet chip technology. And there is a slide in the backup to show how far the lab came and to give an idea what work is still ahead. This is the protocol after the tissues have been treated, and And the tissues are treated for a period of 48 hours of repeated dosing. The first two are to induce the enzyme of the xenobiotic metabolism, especially phase 1 enzymes, and the third treatment is to address DNA damage, which may be immediately repaired. The design is classical to gene tox testing, a solvent control, a positive control, at least 3 concentration of the test compound, each group represented by 3 tissues. In case the standard dosing regime results in negative finding, then it is recommended to do a further experiment in which afediculin APC is added to the culture medium. APC is an inhibitor of polymerases alpha delta, which also have a repair capacity. And that means that with adding a APC single strand breaks resulting from incomplete excision repair accumulates, amplifying the DNA damage, may be said by the test chemical. A gene tox assay is only complete with measurement for toxicity, for general toxicity, as DNA damage can also be triggered as a mechanism secondary to cytotoxicity. In skin, both human native skin and also skin tissues, only a subset of dermal cells proliferate. And therefore, proliferation as a measurement for general cytotox is not appropriate. And proliferation is also not needed in the Comet, as we learned, to either manifest the readout or to monitor the cell cycle. And therefore, the release of adenylate kinase into the culture medium upon cell damage were used as one cytotox measurement to monitor tissue viability in general. And in addition, the intracellular ATP concentration to monitor the energy status of the cells. Both assays are quite sensitive. And while AK adenylate kinase accumulates during the 48-hour of an experiment, the ATP measurement monitors effects which may have arose shortly before sampling. And the good part in that is that the cytotox and the gene tox measurement are taken from the very same sample. This was a little bit theoretically. I like to show you one example experiment from the validation to show you how such a study, especially during validation, has looked like. The study design is also similar, what you may know from gene tox testing. So we started with a solubility study, then a dose range finder was performed to narrow down the dose range for the main experiments. And you see here two main experiments. And for validation purposes, there is mostly an agreement to perform at least 2 main experiments to obtain data on within laboratory reproducibility. You see here the two cytotox measurement in AK in red, and in blue ATP, which are representative for the entire tissue, and then the genotox measurement here for the keratinocytes of the epidermis and for the fibroblast stemming from the dermis, experiment 1, experiment 2. The gray boxes is the historical control. Validity criteria were also set for valid dose groups and valid experiments. So a valid dose group should be characterized by not too extensive cytotoxicity. So AK should not more than double, and ATP should not be reduced more than 50%. There were also validity criteria for the solvent control and the positive control. And last but not least, a prediction model how to evaluate the experiments have been put forward after a transformation of the data, first with an analysis of variance, and then done a test in which single dose groups were compared to the solvent control. But as for genotox testing, also the biological relevance has been considered in terms of dose dependency, reproducibility of effects, and the values, the genotox values should be outside the historical control. And that is what we are seeing here. So this is the first experiment with the standard protocol, and the historical control was not exceeded, neither in the fibroblasts nor in the keratinocytes. But when APC was added in the second experiment, a clear increase in the fluorescence intensity could have been observed outside the historical control. And then the chemical at that time, blinded chemicals, has been considered positive in the expert judgment of the lab. And with that example experiment, I come to the validation. The protocol I've presented was defined, optimized in the first phase, in which also the transferability of the protocol to in total five laboratories had been proven. And then the phases of testing double-blinded compounds started. In a first phase, chemicals, one chemical were tested in 3 laboratories to add information on the intra and inter-laboratory reproducibility. And then on the last phase, a chemical was tested in 1 lab to extend the chemical space. All chemicals were selected independently, different modes of actions, stemming and covering true positive chemicals. So positive in historical in vitro and in vivo data, the true negatives and the misleading positives, which have been reported positive in classical in vitro genotoxic assays, finding which were not confirmed in vivo. And also the statistical analysis was independent. And this is the overview table of the chemicals tested during the validation, which with an expected negative outcome. And you see here some chemicals were tested in the first phase in 3 laboratories, and then later 1 chemical in 1 laboratory. And then here the overview table of the expected positives and the outcome of all studies have been evaluated. First, the within laboratory reproducibility. So those chemicals which were tested in 1 laboratory under the very same condition, those have been compared. Altogether 44 studies, ending up in a quite good within laboratory reproducibility. The between laboratory reproducibility has been analyzed based on the first phase in which a chemical has been investigated in more than 1 laboratory. And again, this ended up in a quite good reproducibility. And 3D Skin Comet is a very good company, because if you check how the in vitro micronucleus, for example, performed, then you will find similar data. And last but not least, we look on the predictivity. So the correct identification of in vivo genotoxicants. This is quite nice. The specificity, so the correct prediction of non-genotoxicants, is very convincing, ending up in a good accuracy of 92%. In parallel to the validation, the Comet assay, the 3D Skin Comet assay, has also been used for regulatory decision making. And I would like to present a case study here of a hair dye. Hair dyes can only used on the European market if an expert panel to the EU Commission, the SCCS, had looked onto the respective data. So, and this is the data for 1 hair dye, basic brown 17, which data package has been shared with the SCCS, the Scientific Committee on Consumer Safety. It turned out that the hair dye was positive in the AIMS, while negative data had been obtained with the in vitro micronucleus test. Follow-up testing resulted in a negative mouse lymphoma and a negative HPRT. And in addition, the 3D Skin Comet assay has been performed, confirming the lack of genotoxic effect, both in terms of both mutagenicity and clastogenicity. And this data package has been provided to the SCCS, and the negative data of the Skin Comet assay, the reconstructed Skin Comet assay, were accepted in a weight of evidence approach even before formal acceptance of the assay, for example, at the OECD. I just wanted to show you briefly the case study of Basic Brown 17. Two additional case studies on hair dye ingredients has been supported by the SEC, by the Comet assay, and were accepted by the SCCS in a similar way, which recommends using both the Skin Comet and the Micronucleus, the Skin Micronucleus, as a follow-up tool for positive results from the standard battery in a weight of evidence approach since 2014. I already touched the OECD, and after the positive feedback from the several expert panels who reviewed the assay, the 3D Skin Comet has been submitted to OECD for the working program in November 2018, and in the April meeting, the assay has been put on the OECD working program to develop a testing guideline. Meanwhile, the data has been submitted to ECVAM for an official peer review of the data package. And we just heard this week from them that the review is ongoing. So we are very much looking forward to the upcoming years and all the things which will happen at ECVAM. If you would not like to wait until the OECD has finished their work, but to already start using the 3D Skin Comet, you could contract studies at Charles River Laboratories. And with that, I'd like to thank you for your attention with the view on all the colleagues who made that happen, what I presented you. And with that, I'd like to hand over to Stefan, unless there are questions we should address right now.
Yeah, there's one clarifying question. All of you, thanks for your questions. I think we'll cover them at the end, but there's one from Valerie that asks, when you showed the sensitivity and specificity calculations, what was that compared against? In vivo results or another in vitro assay? Or
yes, they were compared to in vivo genotox assays and carcinogenicity data. The external expert panel I mentioned were also responsibility to start with a master list to collate chemicals for which relevant in vivo data were available to have a sound database. And from those 32 chemicals had been picked.
Such an important part of validation is the comparator. Okay, well, thank you so much, Dr. Reisinger. We're going to give the floor to Dr. Pfuhler.
Thank you very much. Let me quickly share my screen. And thanks again for the invitation, much appreciated. And hopefully we'll and thanks for all the interest in the audience. Let me quickly, yeah, I'm trying to get it to presenter mode. Here you go. So I'm following on with the Micronucleus reconstructed human skin micronucleus assay, and we'll give a bit of a more detail on the protocols, how this is run, talk about the strategic fit of the assays a bit more. Kerstin has already introduced the way they are used, but we'll focus a bit more on examples as well, because the assay has been used in different scenarios, which I think are interesting for sure. Now, just again, the slide you have already seen, we are talking the dermal route of exposure. So this is more specific, right, than a standard in vitro cell-based assay, which looks at hazard from, I would think, from any type of exposures. Here we are addressing the dermal route, and the micronucleus covers chromosome damage, I mean, as well as numerical damage, so clastogenicity and aneugenic of the spectrum. I bring this slide up just to underpin a comment that was already made too. I think it's very important to understand that the dermal exposure route, obviously there's differences. And one of them is, I mean, you have first, the skin is a penetration barrier, but also a quite metabolically active organ. And I mean, when we started investigating this, we realized there was more limited data out there on skin metabolic competency, and Cosmetic Europe had funded a program to investigate this a bit more. And the paper is cited here that kind of summarizes a series of, I think, a total of 5 papers. And what we found is that actually the skin can do a lot. What the liver can do, I mean, the size of the arrows just in a way represent the direct comparison. So of course the liver, that's its function, is more competent in a lot of things, but the skin is quite good at doing pretty much what the liver can do, except the monooxygenations. Let me quickly change my pointer here. The monooxygenations it's not quite good at, but it's actually inducible. And just to reiterate, that has been an important point, and that's one of the reasons why we went with repeated exposure protocols for these skin assays. I think it's a point to remember. Now, the micronucleus is built on obviously, again, reconstructed human skin tissues, and in this case, actually, matrix tissues. I saw a question in the Q&A box that I can maybe address here because it fits. I mean, we have worked with the matrix epi 200 models. They work very well for micronucleus. They are more simple epidermal models. They have a quite high proliferation rate, which is important for the micronucleus. But they have some other things are maybe not as good as with full thickness models, who have a layer that represents the dermal portion of the skin. And they are a bit more in vivo-like compared to a simple epidermal model. Here, what we get is good division rates, and we need that for the micronucleus because we are only evaluating cells that have been divided. So it's a cytokalasin B based concept. We apply the substance topically. We dose at intervals of 24 hours. We look for precipitation, which has turned out to be an important part, right? If you kind of cover the cells, the skin tissues with your substance, it'll suffocate the tissues and you're getting false positives. I'll show an example of that later. Then you separate the single cells and do a manual scoring and look for binucleated cells that are carrying micronuclei. Detailed methodology information is in the Dahl et al. Paper that also has a scoring atlas for those that are more interested in these aspects. The experimental design in a nutshell, I mean, again, it bases on the OECD 487 Cyto B method that we've modified to suit these tissues. It's 2 or 3 treatments at 72-hour, 48 or 24-hour before you do the workup, a minimum of 3 doses. We have typically 3 tissues per dose, 2 are acceptable, and a minimum of 500 cells, binucleated cells that are evaluated for micronuclei. The maximum dose is 1600 microgram per square centimeter. If the compound is cytotoxic, it turns out, as for other gene tox assays, it's important to capture that dose response, have a cutoff for too high cytotox, which in this assay, like for the OECD guideline method, is 60% toxicity. So in the highest dose, we are aiming at 50 plus minus 10% tox. In the intermediate dose, something around 30%, and low tox is kind of 0 to 20 at most, right? And as toxicity measures other than for the Comet assay, where these parameters can be used, we use binucleation rate, and the cutoff is that you need to be above 40% compared to the control, or cell counts. And really what counts is the more sensitive method. That turned out to be the best concept here. So now I show you the decision tree that we use for the validation exercise, where we had initially started with a 48-hour protocol. And over time, we learned that 72-hour protocol is probably better, and I'll explain a bit why that is later. So we started with this protocol for the validation, a dose range finder, and then a definitive study with a 48-hour protocol. If positive, we are done. The conclusion is positive. If negative or equivocal, we do a confirmatory assay at 48 or 72 hour. And you see already some phases mentioned. I'll quickly show you how the layout looked like, just to show that it was quite complex and there were some iterations over time. Again, if you get a positive response here, you call it positive. Negative, you call it negative. If again equivocal, you would do a second confirmatory experiment, and then again you could still end up in a equivocal conclusion at the end. Now, this is the new design, and that's as per recommendation of the IWGT expert working group. So we would, if we do an assay today, we start with the 72-hour right away. If negative, if positive, we conclude positive. If negative, we concluded negative. There's no need to repeat the assay. If equivocal, we would do a confirmatory assay again at 72 hours and adjust our dose spacing, optimize toxicity, hitting the right levels of toxicity. If negative, again, we are overall negative or positive or equivocal. So that's, if we do an assay today, that's our recommendation also for folks that are using this assay. Kerstin talked already about the validation setup. I just wanted to quickly highlight a few things again. We have had an international validation team with involvement from ECVAM, the European Center for Validation of Alternative Methods, right from the start, and have put the selection of substances in the hands of subject matter experts. One of the problems we were facing, because of course for the dermal route, if we look for compounds, we would ideally be comparing to in vivo dermal gene tox or in vivo dermal carcinogenicity studies, but there's not a lot really available in that space. So the substances in our validation data set for which we know what the outcome would be through the dermal route is limited. But what we would expect is that the oral route exposures typically would more represent a worst case in vivo outcome. We had a steering committee in place that we expanded as needed. and had 6 international laboratories that were experienced in Genotox and in working with 3D tissues involved. Had constant discussion and calibration with the scientific community, and were walking through this as a team through the different validation phases that were mentioned already: optimization, transferability, intra and interlab reproducibility, and then the validation of the coded compounds. So again, here you have some names on the list. I'm not going to go through this, but just to show that, you know, coding and decoding, who did this and which people were involved, because that's important, I think, to judge the validity of such an approach. A quick note about the timeline for the RSMN, for the micronucleus. This looks more complex than it did for the Comet because we had more iterations and had to apply learnings and take a few loops. So I won't go through all these phases and why we did them, but I think it's important to understand why in the example that I've been sharing, you are seeing sometimes more than 2 or 3 experiments for one compound in one lab. So sometimes the experts wanted us to go back and test compounds that have been tested again in a coded fashion with a 72-hour protocol, for example. So the labs had, but it was always coded and steered by external experts. To highlight that the validation outcome is published in a mutagenesis special topic that Joanne Doak has edited. Rafaela Covey from ECVAM and I were the guest editors for that. It includes, I mean, not only the validation papers that I put on the screen here, but also other interesting papers on Skin Comet and Skin Micronucleus. And with that, I wanted to show you quickly a few examples from the validation data set, just to show how the data look like. I picked a couple examples. You will find them in the validation paper. In the supplemental figures is where the detailed data go, because there were, I think, around 300 total graphs in there. And the first one is colchicine. And this is how this is built. On the right hand, you will find the lab mentioned. So the first 2 experiments you see are from lab A. The second 2 are from lab C. You will see here the number of micronuclei on the scale on the left. And let me click once more here because I blew it up a bit so it's easier to read. So on the left-hand scale, you see the number of micronuclei in percent. On the right-hand scale, this is the cytotoxicity measures. The closed bars here are binucleation. The closed squares are binucleation. The open squares are actually relative cell counts. The dotted line up here is the limit, the cutoff level for cytotox. If it goes below that, like in the example here, we didn't score them because we consider that exceeding toxicity. And at the end of the graph, always you see the numbers for our positive control. So you can see, of course, colchicine and aneugen clearly positive in both labs in all the assays that were done. And those were all 48-hour experiments at that time. The second example is a base analogon, sorry, that you all probably know, 5-fluorouracil. And that turned out to be more difficult to catch. So you can see from lab A, like in the initial approach, that the lab actually tested up to very high doses, up to the limit of 1600 microgram per square centimeter. The cytotoxicity curve is actually flat. It hovers around 40–45%, but doesn't kind of exceed the toxicity limits. So those were scored and nothing is happening. And then once I think we realized we missed the middle kind of medium toxicity kind of level, and that was tested again in later phases of the protocol. It came up clearly as positive, more in a mid-level toxicity of like 80, 70, 80% compared to the control. And that was repeated here three times. And even in the 48-hour protocol, once you pay more attention to that, you get a positive response. Lab A, I think that's probably a typo here, lab tested it again in a 72-hour, and it was positive again. Now, when lab B tested it actually at 48 and at 72 hours, and in that lab, they couldn't determine a positive call. So that was more a clean negative for this compound. So that was a false negative response. So when in the context of the validation, so basically, if that was correct, and this was the same lab, then there will be an overall positive call. So that's a correct prediction. Lab B had an overall negative call. That's an incorrect prediction. So it goes in as 50% correct in the evaluation, but for between lab reproducibility, this goes in as a 0, as not reproducible, right, between labs. So to give you a glimpse on the validation outcome, and I don't expect you to go through that. I mean, really, if you're interested in the detail, look at the respective paper. A more detailed citations you will find on the last slide. I mean, that's the overall of more than 40 compounds for this assay. Before I come to the overall numbers, I wanted to stress again how we are using this in a concept that really looks at this in a battery approach, right? So now in cosmetic syrup, and I think in other in the meantime also I think in a lot of other contexts, the two-test in vitro battery has been kind of turning out as a very valuable and sensitive way to go. So you capture all the genotox endpoints by a combination of the Ames assay with the in vitro micronucleus. So that's the concept we're using for cosmetic ingredients, also acknowledged and approved by the SCCS, the Scientific Committee of Consumer Safety. So if we do an Ames and the Ames is positive, we would then do a micronucleus, and if that's positive too, we would have to do both the micronucleus and the Comet skin assay. That is not a typical scenario, right? If that happens, we wouldn't normally put an ingredient like that in the marketplace. If it's Ames positive but negative in the micronucleus, we would look at it in the Comet, Skin Comet. If it's Ames negative, micronucleus negative, no follow-up is required. If only the micronucleus positive, we would test it in the skin micronucleus assay. So now, when I look at the validation outcome for a micronucleus in itself, when it comes to reproducibility, and that's somewhat hampered by the complexity of the design that we have been choosing, but still we get a pretty good within lab reproducibility overall between all the labs of 84%. The between lab reproducibility was short of 80%, 77%. When you look at the overall predictive capacity, we are getting a sensitivity of 75% overall, a specificity of 84%, and an overall accuracy of about 80%. So when you look at that, please keep in mind that when you now apply this concept that we have designed it for, and you look at the fact that really many of the true positives, compounds that are positive in vivo, are double positives in that two-test battery. When you apply that to the overall data set, the sensitivity really increases to 89%. And the overall specificity goes down a little, but is still, I think, at 79% is the overall number at the end. So we get very good numbers when you put it in context of the strategy that we had been really applying here. Now, I think hopefully the most interesting piece is the practical use of these assays. Kerstin already mentioned that the Comet assay is available commercially. That's also true for the RSMN. That's offered in several labs under GLP. And we have worked through, and other people have worked through examples of how these assays have been used in the context of regulatory or internal decision making. Just wanted to mention a Comet example already that was presented by Kerstin. I'm going to show you 4 more brief examples because they are showing the breadth of how this can be applied. The first one, again, is in the context of an in vitro only strategy that was applied for fragrance materials. And the RSMN here was also used as a second tier, higher tier assay in that strategy, similar to what we've done in cosmetics Europe. And this hopefully highlights a good concordance with in vivo results. The second example is again a hair dye, which I think nicely shows the effect of skin-specific metabolism here on in vitro results. The third example is actually a nanomaterial where I think you can nicely also see barrier function. Example number 4 is basing on a recent example based on an aneugenic dermal drug published by Pfizer, kind of having a bit more of a look in terms of hazard identification versus risk assessment. And I'll talk about a few limitations that come in the context of that. What I won't discuss because I don't have time and it's work in progress, but I wanted to mention this. We have a project, Cosmetic Europe has a project together with IIVS, the initial inventor together with us in Procter & Gamble of this method, the RSMN, to establish a photo skin micronucleus assay that would enable us to detect genotoxins that are activated by sunlight, by UV irradiation. So that's work you will see more in the near future on. Example number 1, the RSMN assay is part of the RIFM, and RIFM stands for Research Institute for Fragrance Materials, screening program for genotoxic potential of over 2,500 fragrance materials. That program used the blue screen assay to prioritize for further testing, and then a 2-test in vitro strategy, like same thing, AIMS and micronucleus in vitro was applied. And for those that were positive in the in vitro micronucleus, the skin micronucleus was used as a follow-up. Now, important to mention that many fragrance materials are also used as flavor materials. And the EFSA, the European Food Safety Authority, requires in vivo follow-up testing in the context of positive results in vitro. So for a lot of those, in vivo data exist, and that of course was a good thing for us. So we could compare the results directly with in vivo results here in this study. And there's an aspiration, certainly important to mention, to avoid in vivo testing in the future, also in the context of oral exposures. And there's an assay that's out there that not everybody's aware yet. It's a Henkel micronucleus assay that works with kind of fertilized chicken eggs. Kerstin Reisinger is an expert in that, and there's a couple of papers that are in press in that context, also in a mutagenicity special issue that's in the works. But the manuscript I'm talking about now is in press. You can find it under this link here. It's also listed on the last page again. Now, again here, the table is taken from that paper. I'm not going to go through every individual chemical, but I want you to look at the overall grid. I mean, all of these compounds were actually negative in Ames. You won't see that in the table, but they're all Ames negative, but they are positive in a GLP compliant, OECD compliant in vitro micronucleus. And most of them were positive, some had equivocal response, and we performed 3D skin micronucleus for all of them. And actually they all came out negative in this assay. But we were actually happy to see that for those 19 where in vivo data exists, there was 100% concordance. So even after all exposure at guideline compliant high doses, those were all negative in the in vitro micronucleus, in the in vivo micronucleus. And most of these in vivo tests were also GLP and OECD compliance studies. So just wanted to highlight that comparison exists, and the paper is in press and should soon be final. The second example is from the hair dye arena again. I used to spend quite a bit of time in hair dye toxicology, so maybe not surprising. It's an example we worked on a while ago, but I think it illustrates something quite nicely, so that's why I put it in. So for PPD, paraphenylendiamine, the genotox data situation is as such that it was positive in the standard battery. It was positive for Ames and chromosomal aberration in P53, actually incompetent cells. And the Mars lymphoma was just positive, but it wouldn't be positive according to new criteria. It was negative in an HPRT assay. The SCCS actually assessed it as non-genotoxic because that dye has a lot of in vivo data that is historic, and it was negative in several studies in the micronucleus, in the UDS, in liver, but also in the Comet in 8 organs. So that is, I think, overall was considered a convincing package. Importantly, it was also shown to be N-acetylated pretty much completely when applied to human volunteers in a hair dye formulation. And that's something to keep in mind when we look at the next slides. Now, we have tested it at P&G in the 3D skin micronucleus and got in 2 repeat assays a pretty clear negative response for this hair dye. And again, I mean, it's kind of the same. The closed squares here are micronucleus counts, and this is binucleation and total cell counts. So we were asking ourselves, I mean, here, is it really negative because we are seeing detoxification in the skin? Is there a skin first pass effect here? And the skin, human skin, as well as these skin models, as we have shown in the Cosmetic Europe program I referred to earlier, is N-acetyl transferase proficient. We have done some work on this, and for PPD specifically, and have tested PPD in the Comet assay in different cell lines. We've used native V79 cells that have not very much metabolic competency, so it's a strong positive in the Comet assay. If you use a V79 NAT1 compliant version of the cell line, you're getting a negative response in the Comet assay up to 10 millimolar, up to the highest guideline specific doses. And actually, you're also getting a negative response if you use human keratinocytes, the cell line HACAT, you're also getting a negative. So really, the gene tox is abolished even in vitro when you use these not competent cell lines. So I think we can say the skin probably here has a first pass effect. And that was confirmed in a more recent project, also from cosmetics Europe, where for a higher number of compounds, liver metabolism was compared to skin metabolism using actually skin S9. And when you look at this graph here, the orange crosses here represent the data with skin, the pink ones represent liver. And what you can see is that really the speed of N-acetyl transferase conversion to the diacetylated PPD form is as fast in the skin as it is in the liver. I didn't put that slide in, but they could also show that PPD, the parent, disappears at the same rate as the diacetyl form forms. So I think pretty nice data here to show that in this case here, the skin models are a much more realistic model for the risk assessment in the context of gene tox for dermally applied compounds. Now, example number 3 is slides are courtesy of Shireen Doak, Professor Doak at Swansea University, who worked with skin models also in the context of nanomaterials. And this is from a publication that is cited here. And the picture shows 85 nanometer amorphous silica nanoparticles that are topically applied. And you can see already that at this dose of 50 microgram, you see quite some coverage of the skin surface with these nanomaterials. Now, what they have done is they have compared like results in the 2D model, which was TK6 cells versus the 3D skin models. And what you can see here is that you get a clear positive response when you are using nanomaterials and test them in a standard cell culture model. You're not getting a positive in the 3D model, neither actually if you apply them to the medium or you apply them to the surface. And there wasn't any kind of anything here. There was too much toxicity for this high dose. And basically that's how that dose looked. So the skin was totally covered with these nanoparticles and was suffocating the tissues. So you were seeing toxicity, so no binucleation could be measured or not enough. Now, they did electron microscopic investigation. In TK6, you could clearly find the particles in the cell. Of course, you could kind of trigger inflammatory responses when you get internalization of these particles in the cells. But what they could clearly show is that when you apply the particles to the skin dermally or even through the medium, the keratinocytes did not take them up. And of course, the stratum corneum even more. I mean, there was a total blockage of the nanomaterials on the skin surface. So basically, you're not seeing any exposure, which kind of gives you a better grip on the risk scenario here for these amorphous nano silica materials, and these were even the small 16 nanometer materials. Coming to the last example.
Stefan, I'm so sorry to interrupt, but we have about 15 minutes left in our time. So
yeah, I'll be wrapping up after this slide. Just real quick here, what the group from Pfizer done. Mike Schuler is the first author. They have looked at the RSMN as a tool to do risk assessment in the context of aneugenic dermal drugs. And what the authors could do, which is very nice, they got convincing positive responses for these antigens in the RSMN, and they could rank order them according to the potency of the antigens. So they basically said they could demonstrate that the epiderm is sensitive for hazard identification. But what they could not do was to bridge to a risk. So they really suspected that the result might be different in vivo, and they went to the mini pig. And pigs have, the experts will know that, a more human-like skin. So when they tested these drugs in the mini pig, there wasn't any positive responses in that assay. So they basically concluded it's great for hazard identification, but it's still over-predicting. And that's maybe not so surprising. What they also promoted the use of flow-based alternative markers, which could be interesting to address some of the limitations of the assays, which is really that we have a limited selection of qualified solvents available. That aqueous solvents are problematic, and the solvents we are using, like acetone and ethanol, are forcing penetration. And importantly, the evaluation is quite time consuming. So really automation would be a desired thing for us to do. With that, I come to the summary and go through that quickly. We are using these skin models because they consider the main route of exposure of cosmetics, as well as skin-specific metabolic fate. We think we have successfully validated these assays. And if they are used as intended, we get an overall sensitivity of 89%, an overall specificity of 79% compared to in vivo data between the Comet and Micronucleus. It's commercially offered now, so they are out there. You can find labs that can perform them for you, or protocols are available for you if you want to establish them. We could show that when used for fragrance ingredients, we had a very high concordance with in vivo data again. Could show that they have relevance as an exposure route specific tool in the case of the nanomaterials, for example. And wanted to highlight again that OECD has approved the development of 2 separate guidelines, and that they are formally undergoing peer review by EGRAM, and if successful, which I hope it will be, OECD guideline development can start. And with that, I thank you for attention. Want to make sure I mentioned that a lot of people have been involved in this, doing all this work, a lot of external experts that have helped us with this, and we are very grateful for that. And want to acknowledge some of the key references in case you are interested. They are captured on that last slide for the Micronucleus, and Kerstin had a list for the Comet assay. And with that, I think we are ready for your questions.
Well, thanks to you both for just a wonderful presentation. Wonderful set of presentations on these assays. There's a lot of interest, I can tell. So let's get to some of the questions. So one maybe quick question is, and you, Stefan, you mentioned the solvents that are used, and they're wondering why those particular solvents were chosen. Is it to aid solubility of the test materials, or?
I mean, maybe I take this very quick. I mean, we put quite some effort in it in the beginning. I think it's several aspects. One of them is getting high enough doses applied to the skin surface, because I mean your dose it's different than applying something to the medium. So you need to concentrate your substance quite highly in your application dose, because you can't leave a film of liquid on top of the tissues because they do not tolerate that very well. So we are limited in the amount of solvent we can apply. So solvents that actually are, first of all, better solvents for a broader range of chemicals, but are also evaporating from the solvents more quickly, turned out to be better. So that's why I think we were more limited to things like acetone, ethanol, but we did not put a lot of effort. Once we had decided on those that work, we didn't go back and put a lot of effort in trying to requalify other solvents. I'm sure there's more that can work, and I think people have used others. So there's other options out there.
Right. And let's see, there's another question. This is directed to Kerstin. Did you analyze why some of the compounds were misclassified in different labs in the Comet assay validation?
This was indeed true for cadmium chloride, which was positive as expected in 2 labs and negative in another one. Cadmium chloride is not such an, or was not such an easy chemical to test, and the mechanism, the mode of action of the cadmium salts in terms of genotoxicity are not fully elucidated. When we obtained the results, we looked into the literature and found 2 in vivo Comet studies, and one study showed equivocal results in 2 organs, and the other study showed negative results in several organs. So we did not identify the reason for the negative call in the one laboratory, but we considered it is a challenging chemical to test in a Comet assay.
Okay, great. There's a question about regulatory acceptance, and I know there's a lot that both of you mentioned in terms of ongoing validation and OECD test guideline. Also that there are several labs that are offering both of these assays already, and that there are protocols that presumably people can contact you if they would like them to implement in their own labs. But I wonder if either of you have anything to say about specific regulatory agencies that are already accepting this, this method or these methods.
I think you had seen some examples of us submitting data to the SCCS, yeah, in the context of mostly of hair dyes, and the SCCS has been encouraging us to develop these assays and encourage the use of these assays, so they accept them in a weight of evidence scenario. And I would say that's probably true for other agencies too. And I mean, there's a reason that Pfizer, for example, has used the assay. So I'm pretty sure it will be part of their submission. But I don't think FDA will accept them as a standalone overriding in vitro yet, right? So we are encouraging folks to use these methods in their regulatory submissions, also for regulators to gain more experience and gain more confidence. But hopefully the formal validation peer review will help, and then the guideline development. But unfortunately, this is always so slow, right? I mean, the whole process takes so long. So we think the assays are ready for use and are being used. And there's a reason the lab have established them and offered them. So there's interest out there.
Yeah, I often think that regulatory agencies starting to get results submitted kind of helps the guideline process along in a way, because they are gaining confidence and realizing that they would like this assay to be available, and kind of helps. So great. So another question is related to just wondering about the Het micronucleus that was briefly mentioned and whether that's currently undergoing validation or is there another status?
Yes, I can shortly say something about this. We've finalized the validation, and just last year, 2 publications are out there in mutagenesis. One is on the protocol, a picture atlas for the analysis of the micronucleus. And it's, as Stefan mentioned, it's in fertilized chicken eggs, standardized eggs, normally used in pharma industry for the production of the vaccines. We do analyze the micronuclei in the circulating erythrocytes. And we also finished a validation with excellent predictivity. And if you type in PubMed, RSMN and my last name, you will find those 2 publications and others around. And or you could either contact me directly if there's an interest.
Maybe actually what we could do is add the links into the follow-up email because everyone will get a follow-up email from Zoom with links to the recording, so we can pop that in there too.
Thank you.
Okay. There's a question that, and realizing we have 2 minutes, and there's some of these questions we're probably not going to get to, unfortunately, but there is a question about using the 28-day dermal toxicity study. So using the reconstructed skin, so both of these assays, in place of a 28-day dermal tox, presumably one that is investigating genotoxicity.
Yeah, I mean, the tissues we were working with are not don't survive such a long period. I think you can typically use them for about a week's time period. So 28 days won't work. But there are other dermal models out there and new models that are developing that have a more sustained long life. And in that context, maybe I can quickly tackle another question. I mean, binucleation rate. So for the Comet assay, you could easily do that. For the micronucleus, you need a certain amount of binucleation. And the minimum for us is 25% binucleation over the course of the assay. And I think if you would do a more longer term, that might be a bit tricky because they at one point will stop growing. But if they are healthy, they will still survive for quite a while, but they stop dividing.
And with regard to the Fenian tissue, it has a clearly longer culture time than a week, and you could visit the homepage I put in the slides.
Wonderful. Well, I think that's all the time we have for today. We will, if we didn't get to your question, we'll pull them out and see if we can send you some brief answers. But thank you to Dr. Reisinger and Dr. Pfuhler for coming and giving us this talk. It's been extremely informative. And thank you all for listening and spending your Friday morning or afternoon with us. It was so cool to see people checking in from all over the world. So thanks for inspiring them to do that, Kerstin. Thank you all again, and see you next month.
Thanks again.
Thank you very much
for your interest and for the invite.
Yeah, thank you.
Everyone.